Welcome to Study Loop.
Okay, so 44 has quite a few parts, so we'll try to get through them as quickly as possible. And part A wants us to describe the features of the graph for positive cooperativity, right? So if our x-axis is our concentration of ligands or signaling molecules, that's the concentration, and the y-axis is the number of sites bound. Okay, so number of sites bound. And positive cooperativity has this sort of sigmoidal shape, or like by sigmoidal you sort of mean like an S shape, right? Okay. And so what that implies here is if you take a distance, okay, like a distance on the x-axis between concentration that's kind of short, you'll see where it connects on the graph here a very different, like a large change in the number that's bound, right? And so that the ability for it to have a large change within a short concentration, that's what gives us this positive cooperativity. Okay, and then part B wants us to take a look at negative cooperativity, right? And so the negative cooperativity is going to look a little bit more like this. Okay, and so similarly, if we take a look at within our same realm here, our same gap distance that we used, you can see then that the change in the number bound for the negative cooperativity here is kind of small, right? So even though the concentration is increased, there's not much change in the number bound, whereas for our positive cooperativity, right, the number changed was quite a bit more. Okay, so that's really the sort of major differences there, and with the graph shapes. Okay, and so then we move on to part C, and this is a little bit confusing because part D, there's a typo in the textbook here. Part D gives us the images that are being described in part C. Okay, so it gives us these sort of a bird's eye view of a receptor. So normally we sort of like have our little cell wall and a receptor sort of is shown sticking up, but this one has two. So instead of like a little cup, we're getting sort of two off the side, right? So it's gonna look something like that. But so we're given this image of a receptor at different stages, right? So when there's nothing in it, when it has one molecule bound, and then again when there are two molecules bound. Okay, and so it's asking us to explain the negative cooperation. So why does having more binding not have like a huge impact, right? So why is it that one being bound doesn't allow for more to be bound? And so if we kind of roll back here and think about what's happening inside. So if we have a receptor, okay, so we're going to have a receptor. There's the extracellular part and the intracellular part, right? And once one binds, we can get our enzyme substrate. We'll get sort of reaction one. So reaction number one. Okay, but we have these molecules floating around, right? And so the speed at which one of them is bound is going to be faster than the rate at which a second one finds its way in here, right? Just by sort of probability. There's two open spaces, and one of them is going to be filled more likely than if one open space is filled. But once this second space is filled, we can in fact get a second reaction, right? So what it really comes down to is this sort of idea where if we have our our receptor and enzyme, and there's so we have two open binding sites, and one of them gets filled, right? One of them gets filled. Okay, well, that's going to happen faster than once the other one gets filled, right? Because finding one space out of two is faster than finding one space out of one, right? And then we can get our second sort of substrate on there. Okay, so this rate is faster than this one. Okay, so we're chugging along here and looking at, so we're sort of in part E now, part E, and what would be the advantages in terms of selection to having two site binding versus one site binding? Well, so that sort of comes back to this previous page, right? Having two site binding allows for two reactions instead of one, and so that allows for two reactions on the same receptor. So you have to then produce less receptors, right? So it's less receptors to build and two potentially different or similar reactions that are going to be used. Okay, and then lastly then, so we're no longer in E, but we are going to be in the final portion, part F. Okay, so now we're looking at the binding curves again, and so if we want to go back to our binding curves, they had a few more given here in the book, right? And in particular, there's one with a negative cooperativity but a low K value, right? So this one is also negative, but with a lower K than the blue. And so just a reminder, K is the distribution of receptor-bound and unbound ligands, right? And so when it's small, the likelihood of binding is low, right? So the likelihood of binding here is low binding, even when concentration is high. So the question is, why would that be useful? Okay, and so this is about a form of regulation. Okay, so even if a particular ligand gets overproduced, the reaction in your body won't ever sort of exceed a certain point, right? So even if you were producing way too much adrenaline, you're perhaps it'll keep your, say, heart from pumping so fast that it stops working, right? So it's a form of regulation. Okay, and so that sort of concludes this lengthy multi-part question here on cooperativity. Again, this was all cooperativity.